So for this lab, we are starting to, we're going to look at enzymes, and we're starting to look at some of the more biochemistry concepts of the class. So we'll still have all the same acid-base ionization, neutralization, reactions that we have been talking about. We'll still need to keep fresh in our mind the different functional groups and their reactivity. But now we're actually going to apply them to processes in our bodies that keep us alive, which is pretty exciting. So to start off, enzymes are our body's catalysts. And so what do I mean when I say catalyst? Well, if we look at a reaction coordinate with starting materials and products, what we're going to look at is if we have different energy levels along our y-axis with our starting material, to get them into product form, it actually takes energy. So let's say we have two compounds that are going to react and form products. As the starting material gets closer and closer together and they get in the right configuration to react, it actually takes energy. Their electron clouds are going to need to squish close together and things like that. And so the maximum amount of energy that in the reaction as a whole is the activation energy. And if we have a catalyst, that activation energy is actually going to be lower. So whether our catalyst is some fancy metal from Gen Chem, or it's actually a metal that we're going to talk about at the very end of the term with some of our enzymes and our metabolic processes, the activation energy will get lower if we have an enzyme present. And then the products that form will be, it doesn't include our catalyst. Our catalyst doesn't appear in the starting material or the products, and the energy between the starting material and the products, let's say delta E, that's going to be the same regardless of if we use a catalyst or not. The only difference is this energy difference, the tippy top activation energy, the most energy we'll need in a reaction, it will be different. It'll be lower if we have a catalyst, aka if it's in our body, if we have an enzyme. And so for this class, we're going to be talking about 6 classes of enzymes, and then in lab we're going to look at 2 specific enzymes, amylase and catalase. Amylase takes the starch that we eat. There's a type of amylase that's in our saliva, and so right when you put starch in your mouth, so for example a potato, which is what we're looking at in lab, the amylase will start to break that starch down into glucose. So if we're thinking about polymers versus monomers, starch is our polysaccharide, so it's a bunch of repeating units of glucose, and then glucose is just the monomer, so it's one single glucose unit. Kind of a handy thing for the purpose of lab is we can track this, we can track this reaction, because starch in the presence of iodine will be purple, whereas glucose and iodine will just be colorless. So we should see, if we add iodine to starch and starch is breaking down, we should see that purple color go and fade to be colorless. In our bodies, looking now at catalase, we have peroxides, which can be harmful to our cells. We have a substance called catalase, which will actually break down that harmful peroxide, and it'll break it down into water and oxygen. And so, in the purpose of lab, this actually is a foamy process. The more catalase and hydrogen peroxide you have, the more vigorously it is foaming. The reaction is going more and more. So the catalase reaction that's really being very reactive with a lot of material, it's going to be very foamy. So for the purpose of lab, we can measure foaminess. The height of foam in our test tube, we can measure that to see the extent of reaction. So for this lab, a lot of the information is actually going to be on a Google spreadsheet labeled enzymes. If you look at page 72 in your lab manual, a lot of this lab is you you taking down observations and quantitative data like centimeters of foam from the catalase reaction. And then the write-up will be, a lot of it is going to be on Google Spreadsheet. So just kind of a trick of the trade for what you should put, what you're going to be measuring. There are water baths that will be set in lab for you. So let's say that you want to go and have a temperature of exactly 70 degrees Celsius to measure the level of catalase at a hot temperature. Well, you can use the water baths right in class. So you don't need to actually get out a hot plate and make up your own water baths. And then the write-up is actually going to be on Google Spreadsheet. One thing to think about though is why? Why are we having you look at the catalase reaction at different temperatures? Thinking about what we're talking about in lecture about enzymes, why? Why would we be concerned with the temperature? Why would temperature matter? One thing to keep in mind is that our bodies are naturally at 37 degrees Celsius, and we evolved our enzymes to kind of be at that temperature. So one thing you might want to think about is to predict, well, we're going to have you look at this reaction at different temperatures. What would you predict the reactivity? When would you see catalase really working well, and when would you see catalase not working so well? You know, maybe what would be a temperature that you would predict catalase not to work at? Or what temperature would you predict catalase to work really well at? So on your spreadsheet, you're going to have your lab manual asks for tables, and you know, as long as you're making a table, you might as well make a chart too. Graphing or making charts is just kind of par for the course of being in science, so we might as well get used to making charts now. So you'll want to make a table and chart for your initial test, and so I'll have kind of the template bare bones skeleton on Google for that. And then you'll want to make a table and a chart, a Google chart at various temperatures. So looking at the water baths, I can get ice for you, and you can also, like, the room temperature water bath will just be you putting your test tube in a beaker full of water. Make a table that gives us information about catalase reactivity at various temperatures, and then your lab manual tells you to share your data, so that you will also do on Google Drive. So you'll submit, you or your partner, at least one person from each group, needs to submit their data for the whole class to compare. And then when you do your lab write-up, talk about, you know, the reason why you're sharing your data is to have more trials. So you could say, well, consistently at 70 degrees Celsius, the whole class found da-da-da-da-da-da-da. So the reason why we're having you share your data is so that you just have a lot more trials. And so that kind of makes our results a little more trustworthy when it gets to the point of describing what happened and then thinking about why. So as part of this lab, you're also going to be looking at the pH, and you want, this is kind of like an added inquiry project for you. When you look at the pH, what we're asking you to do is look at the catalase, or you can look at the amylase reaction, and how does it go at different pH levels. So what you'll need to do is make a Google Doc and share it with me, and you'll have to have the purpose, think of a purpose, as well as a procedure for kind of investigating that purpose. So with pH, share that Google Doc with me, and then I'll know what you need. I can go into the stockroom and get you whatever you like, sodium bicarbonate or acetic acid, if you want litmus paper, the pH meters, anything that you might need. And then you can test the reactivity of catalase or amylase at different pH levels. And so you'll need to make a data table, and you'll need to have a place for observations. You might not have too many observations, especially if it's purple or non-purple. It's just whatever is relevant to your lab. A couple things to think about are the number of trials that you want to do and how many different levels of pH you want to look at. And it's really up to you. And then if it's relevant, make a chart. And so you can do that in Google Charts. And you can think about, well, if we have, let's say, three different pH levels, maybe it makes sense to do a line graph. Maybe there is some kind of other chart that might be more helpful. The goal with the charts is for you to actually see what you're looking at, so you can actually see the results. And then when you come to write your conclusion, the chart will actually help you. So with your conclusion, it's the what and the why. So think about what you saw with your pH test and then why. And so all of this, the amylase and catalase initial test should go pretty systematically, especially if you read the lab carefully before class. This should go pretty quickly because it's all systematic. You can have multiple test tubes and multiple water baths at the same time, especially if you're working with your partner. So this should all go pretty quickly. This might be something that you start to think about now, even before you get into lab. So what kind of pH test do I want to do and why? And then last but not least, for our inquiry project, this might sort of stagger into another day, and that's fine. But with inquiry, I've had a lot of success of students actually bringing in something from home. And so you might want to bring something from home and test its starch level, or something from home and test see if you can determine another, so maybe you know for sure that it has starch, but maybe at different pH levels or different temperatures, or maybe there's a third factor that you can think of that might make a difference. Go ahead and test that. So I've had people bring in apples and pineapples. I've had people even look at different enzymes. So with a little internet research, you know, yogurt does some pretty cool stuff. So think about bringing an item from home. You could test the starch level if you already know it has starch or it doesn't have starch. You could, you don't need to look at amylase. You could look at a different enzyme. But I've had a lot of like really cool, really pretty, really interesting inquiry projects with this lab. And so still, even though you've done a lot of spreadsheeting and lab write-ups, still with the inquiry lab, still include the normal write-up that you would. So also include a purpose, procedure, observations, a data table if relevant, a chart if relevant, and then your conclusion. So this lab, it might spread out into a couple of days, and you will have a lot of material that you're giving to me at the end of the day. But it should be good, it should be exciting. Enzymes, everyone eats, everyone needs enzymes to live, so it's a good, exciting start to our biochem section.